The link, YouTube link. Okay.
What's the
இது லைவா போயிட்டு இருக்கு. அங்கேயாச்சு. அப்படிங்களா?
June 17. WhatsApp connect இல்லக்கா சும்மா கனெக்ட் இப்பதான் சார் அனுப்பிச்சேன். அது மேல இருக்குதா வாட்ஸ்அப் கவி உங்களுக்கு அனுப்பிருக்காங்க. எனக்கும் இதான் வாட்ஸ்அப் உங்களுக்கு அனுப்புறாங்க. அது என்ன கனெக்ட்ல பிபிடி கேட்டேன். யூடியூப் லிங்க் எடுத்துட்டாங்க. அது எடுத்துட்டாங்க. அது எடுத்துட்டாங்க. உங்களுக்கு அனுப்பிச்சு குறைஞ்சிட போது யாராவது வாட்ஸ்அப் ஜூலை 7 காலையில்
ஹலோ ஹலோ.
சரி, எங்க ஸ்பெஷல் அட்ரஸ் அக்கா பேசுங்க. ஹலோ சொல்றேன் சார், கேக்குது சொல்லுங்க.
அது ஆன்ல இருக்காமா?
ஹலோ சார். சார் கேக்குதா?
அது சார்ஜ் பண்ணிட்டு இருந்தீங்களா போட்டு? ஆஹா, சார்ஜ்ல இருந்துதான் போட் கனெக்ட் ஆகலையாமா? மேடமோட ஸ்பீக்கர் எடுத்து வந்து வச்சிருங்க. இல்ல இன்னும் ஆள வரல. கரெக்ட் ஆயிடுச்சு. காவியா பேசுறது கேக்குதா?
சார் கேக்குது சார். சார் சார் நீங்க பேசுறது கேக்குது சார். எனக்கு நம்ம பேசுறது ஃப்ரீக்வன்சி கட்டல பாருங்க. அப்ப நீங்க பேசுறது இதுல கேக்குது, இதுல கேக்குது, மேல கேக்குது சார். எனக்கு கேக்குது சார். வால்யூம் நான் இன்க்ரீஸ் பண்ணி பாருங்க சார். நீங்க பேசுறது கேக்குது சார்.
காவியா கேக்குதா? வாய்ஸ் கேக்குதா காவியா?
சார் எனக்கு கேக்குது சார், எனக்கு கேக்குது சார்.
நீங்க பேசுறது கேட்கலமா? நீங்க கனெக்ட் பண்ணல.
ஒரு நிமிஷம் சார்.
நீங்க வந்து பேசுங்க, நான் பார்க்கிறேன். வாய்ஸ் ஆமா சார், ஆடியோ ஹலோ ஹலோ. ஓகே, ஹெட்செட் தான் வந்துச்சு. ஓ, இதுல ரெண்டுமே ஹெட்செட் கனெக்ட். இது காமிக்கலையே. இது ஆஃப் பண்ணுங்க. இல்ல கனெக்ட் ஆகும்.
இது ஆன் பண்ணவா சார்?
ஆன் பண்ணு. கனெக்ட்ல தான் இருக்கு. கனெக்ட்ல தான் இருக்கு. கனெக்ட்ல தான் இருக்கு. இதுதான் கனெக்ட். இதுங்க ஒரு இதுல இந்த பக்கமா இருக்கு. உங்களுக்கு கனெக்ட் பண்ணலையா இன்னும் ப்ளூடூத்தை? வர இது கனெக்ட் ஆயிடுச்சு. ஒரு நிமிஷம் ஜெனி.
No problem, Mama. No
ஒரு
problem.
நிமிஷம்.
Okay.
1 minute. I'll add
Okay. No issues.
இல்லையம்மா, இந்த காமிக்கலையே, கனெக்ட் ஆக மாட்டேங்குது. டிஸ்கனெக்ட் பண்ணுங்க இல்லைன்னா.
இது ஆயிடுச்சு சார்.
ஹலோ ஹலோ ஜெனி, கேக்குதா?
Yeah, I can able to hear you. Yeah, I can able to hear you.
குட்டீஸ் தூங்கிட்டாங்களா?
Not yet. ஒன்னு தூங்கிடுச்சு, இன்னொன்னு தூங்காம விளையாடிட்டு இருக்கு.
மாமி வரேன் முன்னாடியே.
Okay.
ஹாய் கனிஷ்கா.
How do you know? I don't know, sir.
வந்துட்டா மாமி, வந்துட்டா மாமியோட பேசு.
Okay.
Anta Barwan. சரிம்மா அப்போ இப்போ பார்த்துக்குங்க இது
Hello. Hello.
கொஞ்சம் பேச போறேன். Hello, ma'am. Is it audible? Is it audible, ma'am? நான் பேசுவாங்க.
Hi, Vatu.
Hi, mommy.
All the very best.
Thank you.
சூப்பரா பண்ணுங்க. ஜெகன் என்ன பண்றாப்ல?
ஜெகன் வந்து குட்டீஸ் பாத்துட்டு இருக்காங்க.
நைஸ் நைஸ் நைஸ். மாமா கிட்ட ஃபர்ஸ்டே சொல்லிட்டேன் நானு 9:45 எல்லாம் நான்
ஓகே மியூட் பண்ணிட்டீங்கன்னு நினைக்கிறேன். மியூட் பண்ணிட்டீங்க. ஓகே
Okay, Vatu. Carry on.
Thank you. Thank you.
I am with you.
Okay. Thank you.
All my students are there.
Okay. That's great.
Okay. Bye, baby.
Yeah.
hello. இதுல
சார் இதுல கேக்குது
அதுல இல்ல. அதுல இல்ல. இந்த இதுல வருது. ஆனா அந்த ஒரு டிஸ்டன்ஸ் வராதுல்ல. பேச வராது.
Good morning, Kulkarni sir.
Good morning, ma'am. Good morning.
I am Rajalakshmi.
Pardon me, your voice is not so clearly audible.
Yes. Is it clear now?
Yeah, it's bit clear.
Happy morning.
Good morning. Good morning. Good morning to all participants.
நீங்க அங்க இருந்து ஏதாவது பேசுங்க கேக்குதா பாருங்க.
குட் மார்னிங் சார்
அவங்களுக்கு கேக்குதான்னு கேட்க சொல்லுங்க.
உங்களுக்கு கேக்குதா பக்கத்துல பாருங்க
கேக்குதா?
எக்ஸெப்ட் வேற கேக்குதா
மேல அவங்க
கேக்குதா எனக்கு வெல்கம் குல்கர்னி சார் சொல்லுங்க கேக்குதா சார் காமி நீங்க கொஞ்சம் ஐ வெல்கம் குல்கர்னி சார்
Thank you, ma'am. Thank you.
Welcome you, sir.
Kulu.
Yeah,
Here is
yeah.
my-
Yeah.
here is my-
Am I audible, Dr. Dore?
Yes, professor.
Yeah. Thank you.
And Malathi Chini is there, today's speaker. I think you can see.
Yeah. I can see Dr. Malathi. She's on the screen. Good morning, ma'am. Welcome.
Good morning.
He is my friend. மாமிக்கு தெரியும் ஒரு BHU ல படிச்சார்.
Okay. Nice to meet you, sir.
Thank you. Quite obvious the topic when I saw it, it is almost parallelly overlapping on our work also.
Yeah, exactly.
His PhD work is on talking only. He has
Yeah.
done PhD work in talking.
Part of my PhD. Yeah.
வேற யாராவது பண்ணலாமா இதுல உள்ள முன்னாடி பண்ணிடுங்க அதுக்கப்புறம் மாத்தலாம். வேற யாராவது? இல்ல அது நான் போய் பண்றேன் கண்டிப்பா. அவ்வளவுதானம்மா, வேற யாராவது எதிர்பார்க்க? வேற வேற யார் பண்ணும் போஸ்? ரெக்கார்ட் ஆகுது. Recording is there. Enter பண்ணிடுங்க.
एक टॉक दे रहा था.
அன்னைக்கு வந்து போகல ஏதோ ஜெராக்ஸ் எடுக்கிறதுக்காக ஆதார் கார்டு ஏதோ 5000 ரூபாய் தராங்களாம் லோனு. 1 லட்சம் தராங்களாம் குழுவுக்கு 20 பேருக்கு 5000 5000மா. அப்புறம் அவ மரளிகட்ட சாந்தி அக்கா போன் பண்ணிச்சு.
Sorry, Kulu. It's just a technical snack. I made it. Yeah. Adam, start. Kavya, don't take much time.
Okay. Carry on. You're in house or institute?
This is office one.
Office.
So we made a boardroom for our staff members. So
Okay.
principal madam is there, then other staff members.
Okay.
And students are sitting there in seminar hall. Some of the
Okay.
students. So this is seminar hall.
Offline started at your
No,
place?
I just started today only for who are interested. The students who are all interested, they can attend the practical session that way. Just we have
Okay.
started. Just today only we have started.
Yeah, here also what we did is that we made students to have vaccination.
Yes.
One chair, and
Okay.
we need to take little calculated risk. Otherwise,
Yes. Yeah, that's
online
true.
mode, I don't feel there are many complaints from students
Yes.
that the previous vigor as well as intensity, it's not in online.
Yeah. Kavya?
Shall we start?
Yes, definitely.
Thank you. A pleasant morning to one and all who are gathered here. I'm so excited to host this event organized by Sri Venkateshwara College of in association with the Skies. We are organizing on rituals for structure-based.
The mic's off. The mic's back on.
Kavya, உங்க போன்ல தூரம் வச்சிடுங்க. Don't keep it near. ஆன்லைன் கிளாஸ் ஜாயின் பண்ணிருக்கா அவங்களோட எல்லாம் அது மாதிரி கேக்குது. மியூட் others are mute please. SVC continue, Kavya.
A pleasant morning morning to one and all who are gathered here. I'm so excited to host this event organized by Sri Venkateshwara College of Pharmacy through Institutions Innovation Council in association with Discovery Syndrome. We are organizing a third international webinar on ritual screening of lead molecule for structure-based drug design by our eminent speaker, Dr. Malathi Palayam, postdoctoral research scientist, Department of Plant Biology, University of California. I extend my welcome to our special guest. I owe my welcome to all the participants, students, faculty, professors, invited guests from other colleges for your support. I welcome our respected dean, a prominent principal, all our faculties and students of Sri Venkateshwara College of Pharmacy. Before getting into the session, a few instructions to all the participants. Don't unmute your mic during the session. Don't access your video during the session. Kindly post your questions in the chat box. Feedback link will be posted in the chat box at the end of the session. So no separate link will be sent to the registered ID. Please do not communicate later for the feedback link, and e-certificate link will be activated only for 3 hours. Now I invite our dynamic principal, Dr. S. Sarojini ma'am, to give welcome address.
Thank you, Kartika. Happy morning to one and all gathered here. Myself, Dr. Sarojini, Principal, Sri Venkateshwara College of Pharmacy. And I am very much pleased to deliver the welcome address today. On behalf of Sri Venkateshwara College of Pharmacy and on my own behalf, I extend a cheerful welcome to the international webinar. Your presence makes us very happy. The resource from which the God gives is boundless, measureless, unlimited, unending, abundant, and eternal. With the blessings of Almighty and a spirit full of positivity, let us start our today's event. Virtual screening of lead molecules for structure-based drug design, organized by Institution Innovation Council in association with Discovery Syndrome Club. The fragrance of flowers spread only in the direction of the wind, but the goodness of a person spreads in all directions. Amidst us, we have such an inspiring personality is none other than our honorable and respected chairman, Sri V. Ramachandran sir, who had battled against all odds and reached the pinnacle of success. I welcome you, sir. It's my honor to extend a gracious and inclusive welcome to our Chief Operating Officer of Sri Venkateshwara Institution, Dr. B. Vidya Madam, for her continual and cooperative support. I welcome you, ma'am. Creativity is specially expressed in the ability to make connections, to make associations, to turn things around and express them in a new way. I take a great pleasure and privilege to welcome our eminent speaker, Dr. Malathi Palayam, postdoctoral research scientist, Department of Plant Biology, University of California. I welcome you, ma'am. It's my immense pleasure to welcome our dynamic keynote speaker, Dr. Ravindra Kulkarni, Professor, Department of Pharmaceutical Chemistry, Bharati Vidyapith, Pune College of Pharmacy. Well-wisher is not a word, not merely a relationship. It is a silent promise which says, I was, I am, and I will be with you forever. My hearty welcome to our dignified Dean, Dr. S. Duraival sir. Your goodwill, appreciation, motivation, support, and positive vibration has a great impact in molding ourselves for the integral dimensions of the growth of the institution. I welcome you, sir. I wholeheartedly welcome all principals of our sister institution and welcome other pharmacy college principals, professors, HODs, faculties, and my dear students of our college and other college to this great event. I extend my warm welcome to our Training and Placement Cell Officer, Mr. Anandaraj, Sri Venkateshwara College of Engineering and Technology for your continuous support. Once again, I welcome you all for this great event, and it is our pleasure to have you all here. Thank you all.
Thank you, ma'am, for your fabulous welcome. Now I invite our magnificent Dean, Dr. Duraivel sir, to give special address.
Thank you, Kartika. It's already getting time is running off, and I don't want to wait. All the speakers and the keynote speaker, they are having their own work. So anyway, I appreciate and I welcome all the participants to this wonderful webinar. I must be thankful to the resource person, Dr. Malathi. In spite of her busy schedule, she has accepted this this webinar to give a talk. I know that how much hectic she is. Facing the work. I can feel that one. So it's really I must be thankful to her. Then my best friend Ravindra Kulkarni. Yeah, I just taken his. Like invitation, I have given the invitation only just 2 days back. So he immediately accepted for giving a keynote address. So he's a wonderful person. I thank him for joining with us. I thank all the staff members and HODs, special guest, invited guest, everyone to this webinar. I just extend my great gratitude to our beloved chairman, Sri B. Ramachandran sir, who is behind all our endeavors. So I extend all other my thanks to all other participants, other colleges and other people who are well-wishers to our endeavors. So once again, I thank all. I appreciate the organizers for making this event meticulously. I just hand over the session to the organizers. Thank you. Thank you once again.
Thank you, sir, for your energetic opening. Now I invite our distinguished guest, Dr. Ravindra Kulkarni, sir, to give keynote address. Before that, I request our lovable faculty, Ms. Soumya ma'am, Assistant Professor, Department of Pharmaceutical Chemistry, to introduce our distinguished guest.
Thank you, ma'am. A pleasant morning to one and all gathered here. On behalf of Sri Venkateshwara College of Pharmacy, I am very much delighted to introduce our keynote speaker, Dr. Ravindra Kulkarni, MPharm, PhD, Professor, Department of Pharmaceutical Chemistry, Bharati Vidyapeeth, Pune College of Pharmacy. He graduated his Bachelor of Pharmacy from Gulbarga University, Karnataka, and completed his post-graduation in Pharmaceutical Chemistry from Banaras Hindu University, Varanasi. He worked on design and development of P38 kinase inhibitors in his doctoral studies from Kakatiya University, Warangal, and Indian Institute of Chemical Technology, Hyderabad. Soon after completion of his doctoral research studies, he served for National Institute of Pharmaceutical Education and Research as Research Associate, involved in handling classes for Master of Science in Medicinal Chemistry. In the professional front, he served for BLDEA College of Pharmacy, Karnataka, Malathi College of Pharmacy, Hyderabad as professor, and in Sri Vital Education and Research, Maharashtra. He worked for Tetramon Chemi Limited, Bangalore as assistant manager in R&D. He has guided 23 MPharm students and 1 PhD scholars. He has published around 41 research papers in national and international journals. Your smile is a messenger of your goodwill. Fortunate to have you here on this occasion, sir. Thank you.
Thank you, ma'am, for your brief introduction. Now I welcome Dr. Ravindra Kulkarni, sir, to give keynote address.
Am I audible?
Yes, sir. Yes, sir.
Am I audible?
Yes, sir. Yes, sir.
Yes, yes. Yes, Kulkarni.
Thank you, Thank you, the organizing committee members, the management of Sri Venkateshwara College of Pharmacy, the principal, and my close thanks goes to my best friend Duraivel. Almost 1,000 kilometers away from like, geographical distance. However, we close enough to talk daily, and introducing me to drug discovery. Basically, my professional scientific involvement, it is on drug discovery only, and involving all synthetic way, as well as the molecular modeling approach. Now, latest is that we have taken up, especially even I have been now training in programming skills using R as well as Python. I appreciate the efforts taken by the organizing committee to hang up with such eminent and experienced scientists to share the new vistas and avenues in drug discovery. That's wonderful, and it's in today's lifetime. This is very appropriate and pertinent talk on the title. Basing on that only, I feel that all the students who are developing their career in drug discovery, they are very fortunate enough to have such active principal and dean who almost I'll be getting the flares how many such kind of talks they are all arranging for the young scientists, I can say. So such strides which have been taken by the management committee under their encouragement, the principal and dean, to organize such events, it is commendable. That too in such kind of pandemic situation where we can't able to directly interact one to one. But however, despite of all these negatives, still the management, the college, institute, they are organizing such events. It is from my side, it's a great thumbs up. Always in our institute, and while talking to our students, I used to say always one time, we read history books, and from the history there were so many stories we heard. One of them, the great warrior, is Alexander, who had a dream to conquer the world, and ultimately eventually he failed to accomplish his dreams in India itself. But however, in the modern life, you can be Alexander, you can win the world, but not by having a gun in your hand and the bullets in your pocket, but rather if you try to invent and disclose at least some 2 to 3 clinical trial level compounds and give it to industry for their business, then probably you can be modern Alexanders with respect to money with you. So that kind of opportunities, they are existing in the pharmaceutical industry in terms of new drug discovery. But however, having said about the great level of Alexander's story, the path of becoming yourself to the level of current Alexander, it is not so easy. You know very well, almost 10 to 15 years back, one of the disease which we forgotten, or it was in the bookish level, plague, it reappeared in India, and new drugs were sought. Luckily, we had tetracyclines that could able to take care the disease. But in today's life, hardly within 2 years of life, entire world lost almost 4 million people to COVID. At this moment, we don't have a small pill enough to take care of the viral infection. However, we have got compromised vaccines. They are all available entire the world now. Vaccination, it is not that so affordable at this moment to give entire population of world. So this requires one or other corner a drug discovery. It is very important to bring new drug molecule, and before you make out a new drug molecule, there must be one lead. And to invent a new lead molecule among the space of chemicals, around 10 to the power of 60 chemical space, it's not that easy. The way the drug discovery, it is very hard. The in similar way, the lead discovery also, it is so tremendous stress. And to to have such kind of lead molecule which can be taken to clinical level, all sorts of drug discovery technologies you can assume which can expedite the search. Always they are highly acceptable in academia as well as in industry. And one of the technique is structure-based virtual screening or virtual screening approaches for structure-based design. I don't want to enter into domain of Madam. She'll be dealing pretty better than me at this moment. However, in terms of drug discovery approaches with respect to virtual screening, there are two ways you can have it: to the ligand-based virtual screening and structure-based virtual screening. And due to high precision and very robust and successful X-ray crystallographic techniques, we can have multiple number of protein structures. However, still we have to inch towards success and accuracy towards receptor tyrosine kinase protein structure prediction and determination. Such kind of all facilities help the medicinal chemist people to explore new opportunities in bringing new lead molecule, and one approach is the structure-based design. However, I do still appreciate the new lead molecules provided by ligand-based, where you can have multiple structures and you can opt for the synthesis in your wet lab. But the support given by structure-based drug design is enormous. Very much, commendable appreciation we must have to give to structure-based virtual screening methods, which give us exactly how your new molecule is behaving in the active site with respect to their the binding interactions. So such wonderful information, we gather them from both applying structure-based as well as virtual ligand-based virtual screening method, by which you can end up with number of lead molecules. So take some of them to your synthetic laboratory, synthesize them, and then submit them for actual biological activity. So by which at the end of the study, you can get your realistic activity, which can be hinted by the virtual screening techniques. So if virtual screening techniques provided result as well as the wet lab result, they come on the same similar platform, that means your assumption with respect to the working with structure-based design, it's ending up with a successful result. Then take such molecules for clinical study or in vivo studies, then report yourself with a successful activity with in vivo studies, and then report it for your commercialization. That will take the youngsters to a new height with respect to the fame in terms of a monetary benefit they get it from such adventures. Overall, without taking much time, such kind of all activities the new students must have to have, so that at the early age they will be mastering their techniques, and then they can take such mastery to industry and expedite the industry research output. Overall, I appreciate the management, the principal, Dr. Sarojini Madam, and the Dean, Active Dean, Dr. Doraivel, for inviting me to address this August gathering. Without taking much time, now I leave the podium to Dr. Malathi Madam for sharing her experience. Thank you, the organizing committee members, for giving me this space, and I wish you continue such kind of activities in future also. Thank you, one and all, and I stop at this place. Thank you.
Thank you, sir, for your valuable words in this special occasion. Now I request our dynamic faculty, Ms. Revati Ma'am, Assistant Professor, Department of Pharmaceutical Chemistry, to introduce our today's eminent speaker.
Thank you, ma'am. Happy and cherished morning, one and all gathered here. It's my great privilege to introduce our today's eminent speaker, Dr. S. Malathi Palayam, MSc, PhD, Postdoctoral Research Scientist, Department of Plant Biology, University of California. She graduated her Bachelor of Chemistry in Pondicherry University. She completed her Master of Bioinformatics in University of Madras, Chennai.
She graduated her doctorate in bioinformatics, crystallography, and biophysics. She completed her postdoctoral fellowship in National Postdoctoral Fellow, Science and Engineering Research Board in Anna University, Cronbridge, Chennai. She completed her postdoctoral as a researcher in NIH grant, Department of Molecular and Cellular Biology, College of Medicine, University of Kentucky, Lexington, and Department of Chemistry, University of California. She had an extensive experience in protein biochemistry, crystallization, structural elucidation with special expertise in working with soluble and insoluble recombinant protein expression in bacteria and yeast, purification and characterization. And she also expertised in protein modeling, molecular dynamics, and in structure-based drug design. She has nearly published 9 research articles and also deposited various crystal structures in RCSB protein data bank. She has been well trained in extracrystallography, structural biology, cryo-EM, currently working, and various aspects of biochemistry and microbiology, molecular biology. She awarded as Best Posted Award for the poster entitled Current Scenario of Science or National Science Day. She won award for the paper presentation entitled Preliminary Analysis to Target Pyruvate Phosphate Dikinase from Herbacea endosymbiont of Brugia malai for designing an anti-filarial agents. A good orator is pointed and impatient. You are one such. Happy to have you here, ma'am. Thank you for giving this opportunity. Thank you, ma'am, for your fabulous introduction. Dear participants, it is time for your eminent speaker to share her knowledge on today's topic, ritual screening of lead molecules for structure-based drug design. Now, I request our renowned speaker, Dr. Malathi Palayam, to take over the session. Ma'am, please.
Thank you, ma'am, for the nice introduction. And before I start, I would like to thank the convener, Professor Duraivel, for inviting me to give a talk on this. And I would like to thank Professor Kulkarni for giving a brief introduction. I guess he almost outlined my whole talk. Thank you, sir, like giving a brief introduction and giving a glimpse to the students, which is very essential, I felt. And thank you. I would like to extend my thanks to all the organizing committee members, faculties, staffs, and students, especially. So I see large number of students group on this. Kudos to you and thank you. So just give me a second. I would like to share my screen. Just a second. I'm sorry. You able to see my screen?
थोड़ा देखते रहो बस.
Do you able to see my screen? No?
No, ma'am, it's not visible.
Okay. Now, is it visible?
Now it's visible, ma'am. Yeah, now it's visible.
Okay. So good morning, everyone. So my talk is not so deep into each and every aspects of virtual screening, but it will be an outline and with an illustrated example of what I did with this method or with this technique. So virtual screening, by the word, like you have so many number of compounds or libraries that is deposited. Usually the compounds people will synthesize or people will crystallize will be deposited in a public repository database. There are so many number of database available so far. So usually virtual screening starts with retrieving those compounds from the database, and then you do your search, or you do your experimental plan. So it's basically you have thousands of compounds, or more than thousands of compounds, which you retrieve from the database, and then you do step-by-step process. Yeah. So yes. So you might be aware better than me about the drug development process. So it's a very long road to get one drug into the market. So which starts basically planning your research experiment, like you choose your drug, you choose your disease, you choose particular disease, and in the particular disease, you choose a particular target, which might be a protein or an enzyme or an antibody. So you're different, or it can be even DNA. So your target is a macromolecule. Then once you choose your disease and your target, you plan your experiment, you synthesize the compound, and then it has to go into in vitro and in vivo studies. And then it goes to the healthy volunteers. Once you study all the pharmacokinetics, all the pharmacodynamic properties, toxicology, everything, then it goes into the healthy volunteers, and then it goes into phase 2, phase 3, and it should be approved before it is released into a drug into the market. So it is not so easy. It's very challenging process. So for a move from one phase to the other, it's very challenging. The time taken is pretty long, and the cost is too much. Like, it's very high. It's expensive. So sometimes to move from one phase to the other, many compounds may fail, and you lost your time, you lost your money, everything. So what is the route that is followed in industry as well as in academia? I'm not sure why it's not. Yes. Okay. So what is the route now the industry has chosen, or any drug discovery has chosen, is the virtual screening. So in a traditional drug discovery process, so here, sorry. Okay. So the screening of compounds, it's nearly, you will take more than 1,000 or 5,000 number of molecules. So each molecule has to be synthesized before you test those compounds for biological activity. So that itself will take 2 to 6 years minimum. So every compound has to be synthesized, and you should test those compounds for biological activity. But the virtual screening has changed the traditional way. Like, the compound which you have to be screened, like it's not like it should exist physically. Like, even if you can able to design the compound using the computer approach or the molecular model, you can perform your experiment. So it doesn't need the physical existence. So that's the greatest advantage of the virtual screening. So that is one of the greatest advantage. So you don't have to synthesize, and you don't have to wait for that much long time. So it typically reduces your time, and also it reduces your cost, because all the molecules that you synthesize are not successful. So some may fail during your in vitro, in vivo studies, or even in the clinical trials, it may fail. So how these can be eliminated is by the use of the computer-based approach, which we call it as virtual screening. So which typically eliminate the traditional way of screening, by which we reduce the cost and the time we spend for those research. It's not changing. Okay, so this is how the schematic goes. So first, you should choose your disease target, in which disease you want to work, either in cancer biology or any specific, even cancer biology, I'm saying in a broad terms. So specifically, if you want to target any bacterial disease or any disease that is caused by parasites or a virus or yeast, or so you have to choose your target, in which disease is your focus. So first, once you choose your disease target, then you should see whether those disease target or the protein that is involved in that is involved in any metabolic pathways. So the general metabolic pathways. So then you should choose your target protein. So you should choose your target protein and you should see whether it's involved in a metabolic pathway. And if it is involved in any specific metabolic pathway, you should see whether the same protein exists in human as well, whether the similar protein is doing the similar kind of function in humans. Say, for example, if you take any bacterial proteins and you have to check whether the same protein is functional in human as well. So if it is functional in human, if the human being has the same kind of protein structure and function, then you should be careful in designing your drug molecule, because it can be like, it can target, so your goal is to target the bacterial protein, not the human one. So you have to design in a very specific way that it targets only the bacterial one, not the human. It should not inhibit or block the human protein. So it should be very specific when you target the common protein that is present in both your disease target as well as it exists in host as well. Okay, so if you want to get the protein, like you have a database called Protein Data Bank. So where people who work on X-ray crystallography or NMR, or the recent technique which is called as cryo-EM, like using microscopy, like they determine the three-dimensional structure. So these are the three techniques by which we can determine the three-dimensional structure of the protein. So these structures are deposited for public use in a database called Protein Data Bank. So you can type the name of the structure, like if it's like a protein PI3 kinase inhibitor, or what to say, like a protease, or any kind of protein. So you can just type and search and you can retrieve. So all the species, like it's human or a bacteria or yeast or plants, so everything will be deposited. So you can search your target and you can retrieve from the database. So once you have your target protein, next you will screen your compounds. So now here there are different strategies. The virtual screening can be done. And to perform, like how I said, like the protein structures are deposited in a database, you have the database for small molecules, which there are so many small molecule database. The popular ones are Cambridge Structural Database, PubChem, Chem EMBL, and Zinc database. So there are so many databases that is available to the public, where you can retrieve the structure for free. Okay, so you have the structural database and you have the small molecule database. So now you perform virtual screening. So you choose your target. Say, for example, if you doesn't have the three-dimensional structure of your target protein, then you go for the method called homology modeling. So where there is no experimentally determined structure is not available, in such cases, you model your protein. through computational method called molecular modeling. So, so that I will be talking in my later slides. So you have your target protein, you have your lead molecules, you can retrieve the protein from the data bank and the small molecules from the existing database, and now you do virtual screening. So first, I don't know why it's not going front. Okay. So virtual screening, as I said, like you can do, the strategies are ligand based and the structure based. So my topic is now moving towards the right. It is structure based drug design. So what we do once we have the structure. So if the three-dimensional structure is known, then definitely you will go for structure-based drug design. If it is unknown, you try to predict the model of the three-dimensional structure. But in sometimes, like you may not able to determine the structure through even homology modeling, because you can do homology modeling only if there are similar structures that is available in the database. Because say, for example, if you have a protease, say, for example, it's a pepsin. So if you want to model a pepsin from porcine, say, for example, and we have a pepsin from other species like a cow or a human being or any other animal species. And if you want to, you don't have the structure for the pepsin from porcine. So what you do is you take the sequence of this sequence of porcine, and then you do a blast search. Blast is a similarity search. So you do blast against the structures that is available in the database. Once you do that, it will give the similar structures depending upon the sequence you give. Suppose if there is no structures, no previous other structures available in the database, like as I said, like there is no human pepsin is available, there is no pepsin from cow is available. So if there are no other structures available, then you cannot model your target protein. In such cases, you don't know how the three-dimensional structure of the protein looks. And those cases, like if you cannot predict your structure, or if there are no experimental structures available, then you go for ligand-based screening. So ligand-based screening, it doesn't require the necessary or the knowledge of the three-dimensional structure. All you need is your ligands or the molecules from the database. So that is again classified into different types, either similarity searching or pharmacophore modeling. So the similarity searching, or it involves like these two structures are very similar, except the end functional groups. So the more the similar structures, the more it is closer towards its activity. So you can take an existing drug or existing compound that is well known for act as an antibacterial or anti-inflammatory. So, and if you want, and you can perform a similarity search giving this as a base. So the database, if you give this as a query, it's going to, and if you ask the program to search for compounds similar to this, then it will give a list of compounds which look similar to this. Like it'll have an aromatic ring, and more or less it's going to have the same functional groups. So when the two structures look similar, having the same functional groups and same aromatic rings, and with a very little difference in one or two functional groups, the more closer the activity between the compound you are choosing. So with that knowledge, you can screen similar compounds and you can do virtual screening. The next method is pharmacophore search, where these two structures in any way doesn't look similar. but you can see the functional groups of this molecule is similar to this one, like the double bond O here and the methyl group. So the functional groups between the two molecule will be the same. So that's the pharmacophore search. So when you find a compound and when you want to optimize a particular compound, you change, and when you want to optimize the particular compound, sometimes you change the functional groups in order to bind more tightly or to improve the ADMET properties of the lead molecule, or to improve the more specificity, you change these functional groups to increase the criteria which I mentioned. So this is the slight overview of ligand based. Now coming to the structure based. So you need the three-dimensional structure of your target protein. So in any target protein, or if you take any enzyme, it's going to have a binding site specific for a particular substrate. So like if you know the enzyme, it's going to have a receptor. So same example, pepsin, it's going to have, like, it's the protease. So once the protein binds, it's going to chop into pieces. So your protein is your enzyme, and the one that binds to your protease is a substrate. It will act as a substrate. Once it binds to the active site, it's going to cleave into pieces. So where this substrate goes and bind, that's called we call it as the active site. So if you have a target protein, the substrate cannot bind everywhere. It's going to have a specific site, which we call it as active site, or sometimes catalytic site, where the substrate go and bind. And apart from this catalytic site, some proteins may also have allosteric sites. So those sites are called, apart from the active site, when it binds nearby to the active site and it brings about conformational change in the structure, then you call it as an allosteric sites. So you have a target protein and you have a ligand. So you do molecular docking to get the target ligand complex. So when you do molecular docking, you don't do just with only one compound, but more than thousands of compounds you retrieve from the database and you perform molecular docking. So that's the structure-based method. And sometimes, like, we just focus on this active site alone, and you study the nature of the active site, like what's the charge of this region, and how big the cavity is, how big the surface is. So depending upon that, you design a molecule depending upon the cavity. Sometimes it's linear, sometimes if it's too big, you design an aromatic compound so that it goes and bind and covers the surface of the cavity of the protein. So depending upon the cavity, you design your molecule. Okay, so now I'm going to talk about how I did the structure-based drug design for a particular disease target, which I did during my grad school. And again, I want to tell specifically about this rule of 5, because you know your target protein and you know how to retrieve your small molecules, and you perform the molecular docking. So from more than 1,000 of compounds, how would you come down or narrow down to 1 molecule or 10 molecule or 5 molecule? So there are specific filters which filter out the non-specific interactions of target and ligand. So we call computational filters. So the computational filters, it can be like, you can introduce many parameters to make it like to get rid of the false positives. You can introduce so many parameters, and then to make it a precise and concise protein ligand interaction. So the main disadvantage of this virtual screening is sometimes you arrive at false negatives. So that's why, in order to avoid that, people will do consensus docking, which means you don't use only one particular program for molecular docking. There are so many molecular docking softwares available, either free or commercial. So some of the commercial softwares are called Gold and Schrodinger's Glide and MOE, Molecular Operating Environment. So these are commercial versions. Most of the labs or most of the industry will buy this, or sometimes you have Discovery Studio. So these are the commercial ones. So most academic labs who focus on the computational methods or who work on this drug screening will have these softwares. So they buy this, and you have to do molecular docking in different molecular docking softwares, because the scoring function in each software is different, and hence it will give you the docking result. Sometimes will be like, it will choose the same compound as the top, or sometimes it may vary. So if you want to precise, then you can perform the docking in different softwares and see whether you're getting the same kind of results or more closer results for each pores. Or you can do, or if you cannot able to do this consensus docking, people will do molecular simulations. So if you have from thousands of compounds, when you go down to 5 or 10, then all the 10 or all the 5 protein ligand complex is subjected to molecular simulation, where it mimics the natural environment, like how the drug and the protein acts in the presence of water at pH 7.4. So you create an environment where you create a box, you add water, you put your protein and you put your ligand, and you set the pH to 7.4 or 7. That's the normal pH that exists inside our system. So you mimic the environment, how it is in real, and then you perform, like you allow the molecule to move inside the box you created. So once it starts to move, so the protein conformation, so you can see the open conformation, and you can see how the drug molecule or how the lead molecule goes and bind. Sometimes if you define the target size as this, sometimes the molecule may not reach here, but it may stuck in between that. So those molecules, you just get rid of those complexes. So once you do the molecular simulation, you can validate your molecular docking results are true or not. And then you validate the other parameters like the delta G and the hydrogen bond interactions, and how the protein ligand complex behaves in a given period of time. So when we perform this simulation, we do it for a certain period of time, 10 nanosecond or a 50 nanosecond or a 100 nanosecond, depending upon the computational facility you have, because as the time increases, you need more space for the run to complete. So as you perform this, so you need more space. So you can perform like 50 nanosecond simulation, a decent amount of time to analyze the protein ligand complex. And then you study how the protein ligand behaves in a given period of time, whether it comes out of the protein, whether the protein, like if say, for example, you got a nice protein ligand complex, but once you start doing simulation, the molecule just throws out of the protein. If you see such conformation, then it's not a real protein ligand complex. So you can validate your molecular docking results. And apart from this, you have the rule of 5. This is or the rule of thumb, where any given molecule that can act as a drug cannot, the molecular weight of the compound cannot go beyond 500 gram per mole, and the log p value should be less than 5, which means that determines the how hydrophobic it is and how hydrophilic it is. If it's too hydrophilic, like it cannot cross the membrane. And if it's true lipophilic, it's going to stuck between the membrane. So it should act, it should have both, like amphipathic in nature. It should have both the hydrophobicity as well as hydrophilicity. And the hydrogen bond donor and acceptors. So the hydrogen bond acceptors, it should be less than 10. And hydrogen bond donors, it should be less than 5. And so these are the polar surface area, and these are the 5 rules that any molecule should follow to act as a drug. But there are exceptional cases where most of the HIV drugs and most of the cancer drugs doesn't follow this rule of 5. It's because like we don't have a compounds that is more effective which follow all this rule. So we don't have such compounds. So most of the HIV and cancer drugs doesn't follow this, but still the FDA has approved the drugs and people are taking those drugs for the treatment. Okay, so as I said, like I'm going to tell how I choose my target, what's my target and what I did with that. So this is a very small piece of work. It doesn't go into deep, but it will give you an overall glimpse. So the target which I choose, the protein, the disease target which I choose is filariasis. So filariasis, or it's otherwise called as elephantiasis, where many of you have seen people having big swollen legs and they couldn't able to walk properly. So this is a disfiguring and disabled disease. And mostly these disease is called as a neglected tropical disease. Most of the research doesn't focus on this, on the tropical diseases, and one among this is lymphatic filariasis. So this lymphatic filariasis is caused by 3 different species. The parasite, which is Wucheria bancrofti, Brugia malai, and Brugia temori. And more than 160 million people have been affected. And these parasitic disease is transmitted through the bite of an infected mosquito. So this is my disease target. So I choose lymphatic filariasis as my disease target. So inside this, so how it goes, what's the cycle, what's the brief introduction of this disease is that, like, once the infected mosquito bites a healthy individual, the worm or the parasite goes into the human system, where it develops into adults, like it matures and develop into adults. And then once it starts big, like once it reproduces and starts growing bigger, it comes into the bloodstream through the lymphatics. Like mostly these parasites resides in our lymphatic system, and it completely invade our lymphatics. So once it gets bigger in the lymphatic system, it comes into the bloodstream. And once, like again, when the mosquito bites, and once the parasite enters into the blood meal of the mosquito, where it gets into the abdomen, like a thoracic cavity, where it sheds the outer sheath of the parasite, and then it slowly develops into microfilarial parasites. Like it spent 3 larval stages inside the mosquito, and once it reaches the L3 stage, the mosquito cannot can no longer accommodate the parasites because it's getting bigger. The mosquito is small, so once it bites a healthy individual, again it enters into the bloodstream of the host. So this cycle goes. So this is how the life cycle of this parasite goes and infects the healthy individual. So if you see, like, you can ask me, will all the mosquito bites, like will we get the lymphatics? No, it's like there are certain type of mosquitoes that transmits this parasite. So mostly these Anopheles, Aedes, Manzonia, and Culex types of mosquito transmit this parasite from one human being to the other. So the beautiful thing or the boon of this parasite is it harbors bacteria inside its system. So this Brugia malai harbors the bacteria called Wolbachia. So it's endosymbiotic. This is symbiotic in nature. The parasite has the bacteria inside its system, and it's both are symbiotic in nature. And what happens is that, like, the parasite supplies some of the key elements to the bacteria, and in turn, bacteria supplies key elements to parasites for each other's survival. So to date, what I thought is, instead of targeting the parasite, why can't I target the bacteria that is inside the parasite? And when I studied the genomic sequence of the Wolbachia bacteria, I found a specific protein target that does not exist in the host, like the human. So that it can be specific to the bacteria and not to the host. So far, like if you see the antifilarial drugs, so these are the drugs which you could have heard more familiar to you, like ivermectin, doxycycline, or albendazole. These are the commonly used drugs or antiparasitic drugs. These are the commonly used drugs. It's not specific for the lymphatic filariasis. And due to the increase, of course, it is effective, only like mass drug administration is essential. Like you have to take this at a particular time period, like every 3 or every 4 months. And over the period of time, these drugs acquire drug resistance. So how and there is no universal dosage available. Okay, if you take this much amount of the drug or this many milligram is enough to kill this filariasis. No, there is no such thing. So once you get this disease, the legs will get swollen and you cannot walk. And sometimes your private parts get affected, and many of the people doesn't come forward to tell that they have been affected. So it's a very disfiguring and disabled disease. So no one will come forward to say like, oh, I have got this. So this is a disfiguring disease, and it creates a socio-economic problem. So how we can get rid of this is, the one way I thought is just target the bacteria instead of the parasite. And as I said, when I analyze the sequence of this genome, generally, if you go to the National Center for Biotechnology Information, or NCBI, you have the repository of sequence, either the genomic sequence or from the genomic the entire genome of the parasite or the entire genome of the Wolbachia will be solved by or studied by some sort of consortium. So every disease will have a consortium, like cancer or a parasite or any kind of disease. They will maintain some consortium where their main role is to determine the full length sequence, the whole genomic sequence of a particular organism, so that you can know what are the proteins that that organism codes for. And you can do genomic research or a proteomic research. So you can do whatever you want with that, like how we have, it's been completed, right? The human genome, like human genome project. So we have our entire genome have been sequenced. So it's been recently completed, the whole genome project is. So same way, like you have the whole genomic sequence of this is available in NCBI database. You can retrieve DNA, or you can retrieve whole genome, or you can retrieve specific protein sequence. Okay, so I went into that, and when I analyzed those sequence, I found one protein that doesn't exist in human. So that is called as pyruvate phosphate dikinase. So this is a 96 kilodalton protein, and this is involved in glycolysis pathway. Of course, we do have glycolysis pathway, I accept, but we can see what's the difference between the protein that the host has and the bacteria has. So you're familiar with the glycolysis cycle, like once we have a glucose, how it's been break down into a product and how we get energy. So there are different, so at each and every, so as I said, you choose your disease, you choose your target. My target protein, my target protein is pyruvate phosphate dikinase. And now I see whether this is involved in any metabolic pathway. Yes, mine is involved in the glycolysis cycle. So once we have glucose, it's been converted into smaller byproducts, and finally you will have pyruvate. So how these smaller by substrates are made is by the action of enzymes. So at each and every step, the enzymes act on the substrate and it gives the product. And here the glucose has been converted into glucose 6-phosphate by hexokinase, and this has been further converted into further byproducts. And finally, you have phosphoenol pyruvate that's been converted into pyruvate. So here, the conversion of the phosphoenol pyruvate to pyruvate is an like a reversible reaction. So from pyruvate, it can convert it into phosphoenol pyruvate, or it's been vice versa. So this is true in both bacteria and human. There is no doubt. But in human, like in the host, we have two different enzymes to convert this. So the phosphoenol pyruvate to pyruvate is done by pyruvate kinase, and the reversible, like the pyruvate to phosphoenol, is done by pyruvate carboxylase. So this reaction is done by two different enzymes in the host. But in the bacteria, this is done by the single enzyme called pyruvate phosphate dikinase. So this is the structure of the phosphoenol pyruvate, PPDK. So when I searched in the PDB database, there is no three-dimensional structure available. And hence I went with the molecular modeling approach or the homology modeling approach. I use the software called Modeler, where you have programs written in that, and then you should make use of those programs to build a three-dimensional structure of this. So so as I said, like if you want to model a particular protein, first you need the amino acid sequence. So the amino acid sequence, as I said, you can retrieve those sequence from NCBI database, or there is one more database called SwissProt or UniProt, where you will get only protein sequences. NCBI will have everything, but the UniProt or SwissProt is going to have only the protein sequences. So you can retrieve So I retrieved the protein sequence from the UniProt database, and then I searched for similar structures. So what are the similar structures that has been previously solved and deposited in the data bank? I have taken those structure and then align with my sequence and build a 3-dimensional model. So this, you have so many number of online softwares available. You can do it for free. All you need is just the amino acid sequence of your target protein. So once you design this is a 3-dimensional structure, and you can represent your 3-dimensional structure in many different tools. Like I have used PyMol. So this has 3 domains: the N-terminal ATP binding domain, the central Sieveling domain, I'm sorry, the G is over here, and the C-terminal substrate domain. So what happens? To the N-terminal binding domain, ATP will binds. And then here is a substrate binding domain, either pyruvate or phosphoenol pyruvate will bind to it. So say for example, if pyruvate binds here and ATP binds here, the central Sieveling domain job is it just transfers the phosphate from ATP from the N-terminal domain to the C-terminal domain. So that's why it's called as swingling domain. Like it moves from here, like from here to here. It just happens with a change in just phi degree, a change in phi side degree. So if you are familiar with the amino acids and Ramachandran plot, like the amino acids are connected by the peptide bond. So once you have a 3-dimensional structure, so it has a flexible loop where the change in the phi psi will give more flexibility so that it goes into the ATP binding region here, and there is a histidine in this region, like in this domain, where it gets phosphorylated, and then it moves towards this domain where the phosphate is given to the substrate to form the phosphoenol pyruvate. So again, the ATP here will convert it into ADP, adenosine diphosphate. So this is the mechanism, how it goes and how it works. So this particular protein is completely absent, and hence this acts as a drug target. Okay, so I have chosen this domain, and then this is the zoom in. So this is the zoom in view of the ATP binding domain. So this is the ATP binding domain, and the residues which I labeled are the residues that interact with ATP. Okay, so I have chosen this region. So now so this is called the multiple sequence alignment, where these are the sequences of the protein from different parasites, Trypanosoma, Entamoeba, and Clostridium symposium. So these are the different parasites where it has the same enzyme available. So when I did a multiple sequence alignment, I found like the regions, of course, the ATP binding regions are conserved. So the star indicates those residues are conserved in the parasitic proteins. So it shows like the red regions are the one, my active site residues where the ATP goes and bind. Okay, so now I have chosen the disease target. Then I have my disease target protein, and it is involved in metabolic pathway, and I have analyzed, and my analysis showed this protein is unique in bacteria. And one more advantage of why I choose this protein is the glycolysis cycle. The proteins that involved in the glycolysis cycle, like the parasite doesn't have this proteins that makes this glycolysis cycle. Like the whole protein, like enzymes that is involved in glycolysis pathway is missing in the like in the parasite, the Brugia malai. So which means all this is done by the bacteria. I said it is symbiotic in nature and both helps for their survival, each of their survival. So this is even one more finding, like, so if the energy has to be generated, it has to be generated through the glycolysis cycle that is involved in the bacteria. Of course, it can bypass, but this is one more advantage, like how the drug target can be. And when I did a small research in this topic, like PPDK, I found like there are PPDK inhibitors available, but not for the parasites, but for the plants, like C4 plants. So I have taken those ligands as my starting, like a starting schema. Like, so though I chose those compounds, like it's only one, so I chose that compound. I taken that as a base. So I know like this compound is a PPDK inhibitor, but not for the parasite or not for the bacterial PPDK, but it inhibits the PPDK in C4 plants. So I have taken this as my base, and I asked I searched what are the similar structures that are available like this previously in the database. So I use zinc database. So I have chosen all the compounds that look similar to my query. So which resulted in maybe 200 compounds. I have 200 compounds of my similar query. Then I went on with virtual screening, like high throughput virtual screening. So I docked all the 200 compounds through the Schrodinger's Glide, which is a commercial software. So I have done the molecular docking. So, okay, you have a particular protocol to follow if you want to do the high throughput screening. So at the time, you have your receptor. At a time, all the 200 compounds, like one by one, it will search, it will goes and interact, and you have to define the active site region. So I have mentioned this. This is also I have defined, or I have asked the Schrodinger's the docking program to bind only to this region, because the protein is very big. It is around 1,000 amino, more than 1,000 amino acids, like, and the molecular weight is 96 kilodalton. And if you didn't define the region, it goes and search everywhere. Like you have to define the space, grid space, so that it just search in that region. If you didn't define the space, it's going to search in the whole protein, and computationally it takes more time to complete each run. So I have defined only this region, just search only in this region. So that's what it did. And then finally, out of the 200 compounds, I arrived at 50 compounds. So like I said, we have a computational filters. So I have chosen, apart from Lipinski's rule of 5, in order to be so precise, I want my results to be so precise. So apart from the Lipinski's rule of 5, I have set my own filters. So what is it is the hydrogen bond interactions. Like I have chosen the more tight hydrogen bond interactions. Like the more tight, how I define this, or how generally define this, the distance should be, the strong hydrogen bond is like, it should have a linear, the angle between the two hydrogen bond interactions should be 180 degree. Like if it's linear, then it tells that it is a very strong hydrogen bond. So I have set my distance between 2.6 to 2.9, like, 2.3 to 2.6. And then the angle between this hydrogen bond interaction should be above 160 degree. If it's 180, it is more strong. So I have chosen the limit as 160 degree. And next, it should interact only with the active site residues, whether the compound which I'm choosing is interacting with the defined residues which I mentioned here. So that's the second filter. So out of 200 compounds, some compounds doesn't interact and some doesn't interact with the active site residues. Only few were interacting with that, and among the 50 I filter out. I set my computational filter and I manually looked and screened my hydrogen bond interactions, whether it's interacting in the active site or not. And the next, of course, the common way of choosing the best pores or best molecule is like the scoring function, like the scores. So each docking software will give a different score, like a different scoring function. But generally, the docking scores is a combination of the hydrophilic interaction, the hydrophobic interaction, the van der Waals, the electrostatic interaction. So all this have been combined to calculate the score of each molecule that interacts with your receptor. So these are the top ligands so the more negative it is, the more strong the binding. So that's the delta G is Gibbs free energy, it calculates. So you have like the glide energy, glide E model, and glide score. These are the three scores the Schrodinger's glide recommend us to look into the score, and you just look like you can do it once you get your results, you can do it in an ascending or descending order, and you can choose what is the best score. So these are the best scores. Apart from best scores, it forms, like I said, a strong hydrogen bond interaction and a good score. And so these are the best. So I haven't shown you all the 50 compounds, but from the 50, how I come down is through this computational filter, like the docking score plus Lipinski rule of 5 plus my computational filter to achieve this 6 compounds. Okay, so this is the representation showing the molecule that is interacting with the defined active site residues. So if you see, okay, I'm sorry, I didn't mention what is ligand A or ligand B. Okay, so I'm sorry, like I have given ligand A here, but I forget to mention what is ligand A or B. Okay, so it gives, like it shows the hydrogen bond interaction with the active site residues. So this is how it interacts. And further, I took the one that shows, like I took this one and I went on with the molecular simulation, as I said before. So I have done, like when I was in grad school, I don't have a big computational facility. So I ran it for 20 nanosecond simulation. So initially I didn't plan to run, but when I submit the paper, the reviewer asked me, oh, come on, we need a validation. So I performed the simulation study of 20 nanosecond. And then I validated it doesn't come out of the box, but it stayed. And once you see the graph of the simulation, like after a particular nanosecond, if your run is 20 nanosecond, my simulations shows like after 5 nanosecond, it reaches the equilibrium. It doesn't change much, which means it's a tight binder. So further, I was interested in studying like determining the 3-dimensional structure. So as I said, there is a consortium for every organism. So I contacted the consortium that was initially here in USA. So I contacted them to get the whole genome sequence of the parasite so that I can amplify my own target by designing a primer. So if I know the DNA sequence, so I will design a primer forward and reverse so that I can synthesize my protein, and I recombinantly express this protein in bacteria. So that was my plan. But when I approached, and I even contacted the DHL to pick my sample from there to ship to India. But between USA and India, there is a limitation, like it should be like, since it's a genomic sequence, it should be shipped in dry ice. So the dry ice shipment is prohibited from USA to India. So all my efforts failed. So I couldn't able to get the genomic sequence, and I couldn't able to do or crystallize this protein, or I couldn't able to test my compound, like whatever I have done in my in silico studies. So later I chose a different protein where I have the like a recombinant DNA for that. Recombinantly, like we have BM33. So I have worked on that later. So this is a small piece of work. I hope I have given you whatever I can, and with this given time. So if you have questions, and this small piece of work is published, and I get a lot of recognition for this work in many high impact journals like Cell have quoted my paper. So I got like a lot of recognition for this paper. So I hope I have given a glimpse of the virtual screening. If you have questions, and I'm happy to answer. Thank you. Thank you for listening.
Thank you very much, ma'am. I think everyone gained the knowledge in screening of lead molecule and also on filariasis. It is highly beneficial for all of us, ma'am. Thank you once again, ma'am. Now I invite creative faculties of SVCP, Mrs. Nirmala ma'am and Mrs. Kasturi ma'am for Q&A session.
Ma'am, you're muted, I guess.
Nirmala ma'am, your voice is not audible. It is not audible, ma'am.
Kavya, maybe அது disconnect ஆயிருக்கும் நினைக்கிறேன்.
Okay, okay. Shall I continue?
No, it's not audible. Yeah.
Yeah.
Or you can leave a chat. You can leave a chat. You can leave your question in the chat.
It's already there. They'll ask Chini.
Okay.
Now the session is open for Paris. Participant, please post your question in the chat box, and which will be clarified by our speaker.
There are
your questions in the chat.
Yeah.
Okay. I will go from one by one.
Can I read it for you?
Yeah, no, ma'am, I can able to see. No, you don't have to read it. So I see like what are lead molecule and how they different from the drug molecule. So the lead molecule is the one that you find like that is interacting with the protein, and say for example, from this study which I have shown, I know like the molecule which I have studied is interacting through in silico studies, but I need to validate this through real experimental method. Like I have to study the binding interaction between my protein and the molecule which shows it is a lead molecule. It can be a drug. So you have to perform the binding studies first and see whether you can see the interaction, the real interaction between the protein and the ligand. If it's interacting, what is the binding constants? what are the parameters? What is the dissociation constant? What is the association constant? So you need to derive all the thermodynamic parameters, which tells how specific it is and how much, like how minimum you need. Like the constants give you a concentration, like that is the minimum concentration that requires to inhibit this enzyme. So you need to find that. So you can able to do that only if you have your protein of interest that can be expressed and that can be purified from other bacterial proteins. If you express that in bacteria or yeast or mammalian cells or insect cells, so you need to you use recombinant DNA technology to express the protein, and you should purify from rest of the other proteins. And then once you purify, you need to either synthesize the molecule which you say as a lead, or if it's available commercially, you buy that and you perform a binding studies, and then you study the interaction and you derive the binding constants. So if it should be in a nanomolar. So generally, if you observe the inhibition in a nanomolar, then it means it is highly specific to that protein. And you can also, if you see similar proteins, if a similar protein also exists in human, you can also study the binding interaction of that with the host. So what is the binding constant for the disease target and what is the binding constant for the protein that is in the host? Then you can determine how specific it is. So once you do this, once you find the interaction, then it should went to the clinical trials. And or it is not that easy, as I say. You need to optimize certain functional groups to make it more specific. And once you do all optimizations, and then it goes into the large scale synthesis of your lead molecule, and it goes to the clinical trials. And after that, it's a long way to be a drug molecule, and it should be approved. I'm not sure who asked this question. Does that make sense? Okay. Can Wolbachia take ATP generated by human cells, and why it only depends on bacteria? Okay, so the Wolbachia is bacteria, and that's a good question. Like if it is inside the host cells, and why it should certainly depend upon Wolbachia, the parasite, not the host cells. Like initially, like for their survival, it has to it requires ATP only from the bacteria. Like, so the parasite is the Brugia malai, but the bacteria here is Wolbachia. I'm not sure whether it can, once inside the host cells, it can take up the ATP from the host. I'm not sure. I don't have any idea about that. Maybe it does, or it may not. Like it can only depend upon the bacteria. But that's really a thoughtful question. Thank you. And can we design an ATP analog which, if it enters in human cell, will give energy, and if it enters bacteria, should act as inhibitors? Okay. Again, I have got an interesting question. ATP analogs generally we won't prefer, because in the host, like the humans, we have different ATP binding domains. Like most of the kinases we have in our system will bind to ATP. You know, like kinases, it requires ATP. Without ATP, the kinase cannot be functional. And most of the ATP analogs is going to bind with the kinases. So we it won't be a more specific if you choose an ATP analog. That's what I'm thinking. So if you want to design an ATP analog, maybe you have to design more specific to the Ulbeckia. That is a big hustle, I feel. It's not so easy, as we have most of the ATP binding domains. Like ATP analog is a very generalized thing. Many of our proteins, it's going to interact with ATP. So if I'm in your position, I don't go with this idea. Can we have your contact mail ID? Yeah, sure. I can share my mail ID. Maybe I can leave it here. So I'm leaving my email ID here. Of course, you can ask for help. Like there are so many scholarships available if you want to pursue master's or PhD or a postdoctoral program. Some you can approach directly to the institutes, or like if you have a specific person in mind, like there will be each and every field, there will be one famous person in crystallography, or a genomic person, or pharmacology. Like there will be one big shot available in each university. So if you want to contact them, you can directly email, or you can look for their fundings, and you can join as a master student, or you can do PhD. Like for doing PhD, you need to clear IELTS. So you should have the English proficiency to know, and then you can, so each and every university applications are different, but there are so many scholarships, more than 500 scholarships for pharmacy alone to pursue your higher studies. Especially you have Fulbright scholarship for Indian students. You can do PhD, or you can visit USA as a visiting scholar to do a part of your PhD studies, or you can, after completing PhD, you can come here as a postdoc, and that's what you come as an exchange visitor to pursue your higher studies. And is there any database especially for natural products deposited specifically for natural ligands, not the synthesized molecules? I'm not sure whether that is specially exists for natural products, but I know like it's a common thing so far. Up to my knowledge, what I'm thinking is there is no special thing for natural products, but all the synthesized, because even if it's a natural products or synthesized, you need to crystallize the structure. Once you crystallize the small molecule, you will deposit in a small molecule database. So I don't think we have a special database for natural products up to my knowledge, but if I find anything, I can pass the information to your dean, so you can ask from him. How to module a three-dimensional structure for a ligand molecule, how to learn it. Yes, there are many online tools to draw the three-dimensional structure of a molecule. Like you can just Google search it, online tools to draw a small molecule. It'll give you n number of list. And you have a program, like you have a software called ChemSketch. I can type it here. ChemSketch, that's the software package where you can download and you can draw the molecule of your interest. And then ChemBank, I guess. Yeah, ChemSketch, ChemBank, and for the ChemBank, I guess you have a ChemDraw. Yes, ChemDraw is the one. So you have an online tool as well as you have a package you can download and you can draw. So whatever you draw is in one dimension. You need to optimize to become a three-dimensional. So you cannot whatever you draw as a 2-dimension. So you need to convert into 3-dimension to do molecular docking. So you can do in PyMol as well. You can do PyMol, you can do ChemSketch, ChemDraw, and there are so many online tools to draw your small molecule. Generally, like in all the software packages, you have at the top, you can find help. So if you go to help, it will teach you how to draw a structure in the ChemSketch or a ChemDraw. So you can make use of that and you can draw. It's very simple. It's not complicated. It's just a straightforward. Studies for synthetic compounds. If you like to do, please let us know. Okay. So the enzyme kinetic studies, yeah, I like to do, but I'm not allowed to carry compounds because I need to ask my boss, because I work in a lab under a particular supervisor. My postdoctoral research is under a particular supervision. So before I get compounds and I get any protein from you, I need to ask the permission, because here I am working on 5 different projects. So to complete this, you don't find enough time. So maybe I can ask my friends if I can just direct to them and they can help you. But I'm sorry, I cannot do it. If we want to do research in researching platform, how do we pursue and what to pursue after UG? Okay, you can, I'm not sure what are the different departments you have in pharmacy. You can choose whatever you feel or whatever you are interested in, like a specific department or specific field, and you can search whether that department is available in any country you want to pursue your higher studies. Generally, it will be here, everywhere in the world. So you can choose different, maybe specific, you can just choose 2 or 3 universities. And under the 2 or 3 universities, you can go and see because under same department, you have different professors work, and you can look into the profile of each scientist, and whichever you find is interesting, you like, and you want to pursue, you can directly write an email to them. They more like welcome students' email, like some, if they want to hire any students. So once you complete UG, you can come here as a junior specialist. Like in India, like we have to complete masters to pursue PhD, but here you don't need that. Here, once they complete their undergrad, they work as a junior specialist for a few years, and then they join grad school, like they pursue PhD for 5 years. So here, masters is not so necessary to pursue PhD. So if you complete UG, you have to do your English proficiency test, and you have to clear that, and then you can apply for a scholarship, or you can approach a specific scientist. If you have a specific university like Stanford or MIT or Cornell University or approved university, so you can look into each universities and you can look whether your field of interest is available, your department is available, and you can look into specific scientist. There will be so many, like under one department, you will have 5 to 10 scientists available. Like you can look into each profile, whichever you feel so interested, you can email them or you can contact them directly, or you can look for particular scholarship. If you want to do a PhD program, and you have to write a proposal. So before the proposal, the host institute should accept, like they should give acceptance letter to you. So if you can look into Fulbright scholarship, so they will have all eligibility conditions and requirements, what you should do before you are selected for this. So you can follow that. So each scholarship varies. And there are university, I saw like Cornell University, Purdue University, specifically they are giving for pharmacy. So that's the scholarship, like each university offers under pharmacy department. You can also take a look into that. You can just Google search scholarships available for pharmacy for Indian students in USA, like general Google search. It showed me like 500 different scholarships for pharmaceutical students. So you can make use of that. Yeah, I guess if you have further questions, and I'm happy to answer.
Participants, if you're having any other questions?
And if anything is there, you just make it in a mail because no, she is already running short of time. She is it's midnight there at US. No, like we are nicely sitting here at daytime, and she has started at 10:30. 11 o'clock at 11 p.m. at midnight, and go on giving a lecture. See, we have to take that consent. So please, if any more questions, please put it in the mail ID. You will be get clear. Thank you, Dr. Malathi. So it's really, it was a wonderful talk. I think even we have got this discovery club, discovery syndrome. We are planning to give a workshop to the students how to draw molecules in chem sketch. I think our staff members are planning for that one. So in future also we may be in need of clarifying some of the things. Our staff members also in touch with you to make things a better way. And thank you. I think I'll just Kartika, please take over.
Thank you, ma'am, for taking time to clarify and sharing your valuable words and knowledge. Now, I request our unique faculty, Mrs. Gayatri, ma'am, Assistant Professor, Department of Pharmacy Practice, to deliver the vote of thanks.
Thank you, Mrs. Kartika. Gratitude is a powerful catalyst for happiness. It's a spark that lights a fire of joy in your soul. Happy noon to one and all. Honorable Chairman, valued excellencies, dignitaries, professors, faculties, and my dear students, I deem it a great honor and privilege to propose vote of thanks on this event. I, Mrs. Gayatri, on behalf of Sri Venkateshwara College of Pharmacy and on my own behalf, extend a very hearty vote of thanks to all for gracing your precious time with us. Our first and foremost thanks giving goes to the Almighty, the Creator of all, for what we have and what we don't have. Today, my words are not enough to express the gratitude. I would like to thank our Honorable Chairman, Sri Ramachandran Sir, for his continuous support and guidance that he has extended to all of us. I extend my warm thanks to our vigorous Chief Operating Officer, Sri Venkateshwara Group of Institutions, Dr. Vidya Ma'am, for all the words of wisdom that radiate a source of energy within us. My heartfelt thanks to our esteemed speaker, Dr. Malathi Palayam, who despite her busy schedule had found time to share her experience. Thank you, ma'am, for enlightening us with your knowledge. Today we have the opportunity to hear from you, and it was full of information. It gave deep insights into the topic, and I'm pretty sure that this will help us in our future and in also our research work. My deepest appreciation and profound gratitude to our distinguished guest, Dr. Ravindra Kulkarni sir, for his keynote address, which briefed the entire session. Thank you, sir. I extend my special thanks to our dignified Dean, Dr. S. Duravel sir, for his special address and for being our backbone in our endeavor, constant motivation and inspiring us. I place on record my deep sense of gratitude to our principal, Dr. S. Sarojini ma'am for her welcome address and for her continual support, who work hand in hand with us, which makes us feel relevant. I extend my sincere thanks to all the valued excellencies, principals, HODs, professors, faculties of various other colleges who has joined with us for this event. Thank you all. My sincere appreciation goes to our training and placement officers, Mr. Anandraj sir, Ms. Kavya ma'am, and Ms. Meenakshi ma'am, who are the pillars of making today's event a resounding success. Behind every successful program be the manifold attempts of our committed faculties. I would like to take a moment to acknowledge the immense contribution of our dedicated faculties. Thank you all. I owe my heartfelt thanks to our technical staff for their support. Thank you for being with us. And I would also like to extend my wholehearted gratitude to all the students of Sri Venkateshwara College of Pharmacy and also to the students of various other colleges who have actively participated in this event. Once again, I thank you all for joining this virtual international webinar to make it a memorable one. Thank you all.
Thank you, ma'am. Thank you all once again for your support. We will catch you again on the next webinar. Thank you.
Unmute. Thank you, Chinny. I
Thank
will
you.
catch you later. You
Okay.
just go to
Yeah.
bed.
Yeah.
Take care.
Thank you,
Take care.
Auntie ma'am.
Yeah, thank you, ma'am.
Even
Thank
a
you.
wonderful session, wonderful.
Oh, thank you, ma'am.
Very interesting. You have been involved. I feel to do another research one.
Okay.
That way involvement in the night time you have been spending. Really, it's very great.
Oh, it's been usual, ma'am. It's usual for us to sleep by 12. Yeah.
I'm
Thank you, ma'am.
Rajesh's classmate.
Okay.
Both of us are friends.
Okay.
Malathi College.
Nice to meet you, ma'am. Nice to meet you.
Very happy to
Yeah.
see you. Very happy.
Yeah. Thank you, ma'am. Thank you so much.
பிரியா ஜாயின் பண்ணான்னு நினைக்கிறேன்.
Okay.
இப்பதான் பார்த்தேன் நானு.
Okay.
Okay. Okay, Jenny.
Night time you are spending.
Thank you. Bye-bye. Yeah. Yeah. Bye, everyone. Yeah. Thank you.
So please, participants, note down the link, feedback link, so that you can get a e-certificate. Later on, don't call or don't make any communication through mail, because we are not going to encourage that. So the link is there in the chat box. Please note it down before we are going to end this meeting.